So I wanted to thank everyone again for this award, and I'm very excited to talk to you about my research. Oh, okay. So my talk is entitled A Tale of Two Genomes, Mitochondrial Nuclear Interactions. So everyone here has had the talk and knows how babies are made. The father provides a sperm with half of his DNA. The mother provides her egg with half of her DNA. And then 9 months later, you have a baby. Something that is overlooked in this process is that the mother actually also provides her mitochondria in the egg. And each of these mitochondria have their own separate circular mitochondrial DNA. So, as I said, it's a very small piece of DNA, and it's circular, and it only codes for 13 proteins. These 13 proteins are very important because they have to work with proteins from the nuclear genome to make ATP. And this is the energy that the cell uses for its everyday functions. So this process is extremely important, especially in energy-hungry organs like the heart and the brain. Mutations in the mitochondrial genome affect this process and manifest themselves as mitochondrial disease. And so inherited diseases caused by mitochondrial mutations affect about 1 in 10,000 people. And so mothers who have mitochondrial diseases obviously do not want to pass them on to their children, since they provide their children with the mitochondria. So experts in the medical field have come up with this new process. It's called the 3-parent procedure. And so the father provides his nuclear genome, the mother provides her nuclear genome, but her dysfunctional mitochondria are actually replaced with another woman's healthy mitochondria. So this begs the question, are all healthy mitochondria created equal? So the proper communication of the mitochondrial and nuclear genes are essential to the energy-making process. And this communication has been fine-tuned through evolution. And we can assume that pairs that do not communicate properly have been weeded out by evolution. So we wanted to see whether this energy-making process is affected by changes in the source of the mitochondrial genome. So in order to do this, we look at a population of this cute little fish over here. And they actually, in one population, there are 2 different types of mitochondria, which we call north and south. And so if they're all living in the same population, then they're interbreeding. And if they're interbreeding, then there's mixing of nuclear genomes that did not necessarily evolve with one of these mitochondria. So using next-generation sequencing, we sequenced the genome of 155 of these fish to look at genetic variation. So are there differences in nuclear genes between the fish that have these different mitochondria? So what we found is that out of 11,700 genes, 349 nuclear genes differ between individuals with different mitochondria. So along here, we basically have individuals. And so these are the individuals with the north mitochondria, individuals with the south mitochondria. And you can see that most individuals with the north mitochondria, the all blue ones, so each line is an individual, have nuclear genes that are associated with it. The ones that are mixed, so the red lines, have mixed nuclear genes. So we wanted to know, are the individuals with mixed nuclear genes, does it affect the energy-making process? And the answer is that yes, it does. So all the way to the left, we have individuals with the north mitochondria and the north nuclear genes. Up here, we have individuals with the south mitochondria and south nuclear genes, which are significantly different, and we have evolutionary hypotheses as to why that is, but I won't go into that. But the important thing to notice is that individuals that have mixed nuclear genes actually fall in between. So having different nuclear genes does affect their mitochondrial activity. And I should mention that this was actually done in heart tissues of the same fish that were sequenced. So now going back to this question, are all healthy mitochondria created equal? Well, the answer is no, they're not. But now the question becomes, are they all good enough? So obviously, if you wanted, if we had the choice between having mitochondrial deficiencies or not, you would pick the latter. There's so much that the mitochondria influences in the cell. And there's a lot more research to be done about mitochondrial nuclear interactions and how it affects human health. But it's very exciting to see the strides, the medical strides done in preventing and treating mitochondrial disease. And with that, I'd like to acknowledge my lab members and my collaborators, TASSA, of course, my funding sources, and my institution. Thank you.